Thank you.
Yes, the rest of the students, switch on your videos. All right, so students, as far as the topic we discussed in the previous class, did you people have any doubts in transcription process? Is
No,
there
ma'am.
anything that troubled you? Anything you want me to, you can say, go over again? All of the factors and everything, they were clear? All right. Okay, so tell me one thing, students. The process of like the step that is known as elongation. Now, during elongation, nucleoside triphosphates are added, right? Now, tell me one thing. Does this happen in a template dependent manner or the template independent manner? What is the manner of this happening? Hmm? Does this happen in a template dependent manner or template independent manner?
Ma'am, can you repeat?
The process of elongation during the transcription, the nucleoside base, or you can say the base pairs, they are added simultaneously by the RNA polymerases. So does this happen in a template dependent manner or template independent manner? First of all, you people tell me, did you get the question?
Yes,
How
ma'am.
many of you did not get the
No,
question? How many
ma'am.
of you got the question? Hmm? So what is the answer? Tell me the answer.
Template dependent manner.
Template dependent manner, exactly. It will happen in the template dependent manner only, because without template, how will the whole entire new RNA will be synthesized, isn't it? So yes, it happens in a template dependent manner. Very good. And where are you, all of you? Like, I can see only one student.
Ma'am, 1 minute.
Mhm. All right, so okay, so let's just quickly revise about the transcription, because many a times they will ask you to write about transcription in depth. Okay, so we know that the process of transcription is the second process during the central dogma of molecular biology. Now, in that, we have seen that if we are talking about the prokaryotic organisms, then the process of transcription, it takes place in the cytoplasm. Whereas the eukaryotic cell, where does it take place? Tell me,
Ma'am,
where
can you
does it?
repeat the question again?
The process of transcription in eukaryotes takes place where?
Nucleus.
Site.
Nucleus, yes, nucleus. But in the case of prokaryotes, it takes place in the cytoplasm. The process of translation, that takes place in the cytoplasm of eukaryotes as well. Now, the process of transcription is the process through which the mRNA, or you can say the hnRNA, is formed. Now, what is the common name of hnRNA, students? Hmm? The common name of hnRNA. See, this is not a test. I just asked so as to know where you people are in terms of your understanding of the topic. So tell me, hnRNA. You will remember anything about it or no? No. It is the precursor of mRNA, right? Okay. All right. So, yeah. So now during the process of transcription, we know that the transcription takes place with the help of the transcripting enzymes. Now the transcripting enzyme, which is present in the case of prokaryotic organism, is DNA dependent RNA polymerase. Because why? Because the whole entire process is template dependent, meaning the DNA that has already been existing, the new RNA or the complementary RNA will be made because of that. Now, do you people remember the complementarity question that I gave you in the class? This one? Do you people think now you can solve it?
Yes.
How many of you
Yes, ma'am.
practiced this when you went back home? Like went back home, you are at your home only. I mean, when the class was over. How many of you practiced it? Even if you did not practice, meaning how many of you got the gist of it, like how you were supposed to do it?
I understood.
You got it? What about the others?
50-50, ma'am. I have to practice
my
it
God.
again.
You got it, Ikra? Okay.
Yes,
Khadija, what
ma'am.
about you?
If I practice it once, I'll get it.
Okay, you'll practice once and you'll get it. Okay, what about the others? Amna, what about you?
Yes, ma'am.
You've got this? Like how you're supposed to make a complementary sequence for DNA and RNA?
Yes, ma'am.
Okay, so everyone has got this, right? Good to know. Okay. All right. Now, see, this kind of question, it was like this question came in NEET also 1 year. Okay, to find the complementary sequence of mRNA, this was asked in the NEET examination as well. So this is very basic, very easy, teeny tiny question. Everyone must know how to do this. Like this is the most easiest, and you can say the favorite part of this particular chapter. It's so easy to do so. Now, apart from that, let's come on to the process of transcription. So the transcription, it takes place in 3 steps. These steps are same in prokaryotes as well as the eukaryotic organism. Talking about the initiation, so the RNA polymerase, it binds to the promoter and initiate the transcription process. Then comes elongation. The RNA polymerase, they use the nucleoside triphosphates as the substrates. Nucleoside triphosphates. And what does it do? It polymerizes. Polymerization means you can say the extension of it. So that more and more base pairs are added depending upon whatever is the template of the DNA. And it is template dependent. You people understand what is the meaning of template dependent? How would you define template dependent?
What is template dependent?
Hmm? Yes, say. Yeah, what is template dependent? Are you asking me or are you asking your Google? No shortcut, no shortcut, no shortcuts in the class, students, please. Okay, see, template dependency, that is why I'm constantly asking, do you people understand this? Students, tell me one thing. Do you hate this
Ma'am,
chapter even more than botany chapters?
yes,
Like,
ma'am.
tell me, yes, ma'am.
Genuinely hatred.
Yes, ma'am. It's too difficult, ma'am. It shouldn't be the biology. It shouldn't be this difficult, but it's why is this chapter difficult?
It's difficult for you, for all of you.
Yeah.
Is it because you people are studying it for the first time, right? This chapter has been done in your school?
No.
No, for like none of you. Okay. Anyway, see, students, I understand. Even I used to find it difficult. It's normal. But the thing is that you people are not even trying. You're not even giving 1% of effort. You're like, okay, we find this difficult. We're never going to study it. You know, molecular biology is that part of biology that is even asked in master's exam. Masters. Imagine you people are in school right now, not bachelors, masters, whenever a person goes to do any kind of PG. So in the entrances, molecular biology consists of the major weightage, which is very good for those people who know it. But again, extremely horrible for those who don't know it, right? So what is the problem that is arising? You people tell me, we'll go like that only. Because the next chapter that is to come, human health and diseases, all of you are going to be so in love with that chapter. I already know it. You people are going to pay a lot of attention. I know that. So just think that I am giving you the benefit of the doubt to be acting in the most rebellious way in my classes recently because you don't like the chapter. I'll give you a tiny bit of the doubt. But tell me, how will you go about it? Also, first of all of you switch on your videos. Oh God. This, like, my class takes maximum of the time because of this particular reason. I want to see all of your faces. Come on. Because when I'll see your faces, I'll understand better. But the look on your face tells me everything. Amna, you also switch on your video. And Mariam Lohani, you also do. I've never seen you in the class, not even once. I was checking your paper today. Also, talking about the paper. How was your test, students? How was your test? And many, like a major amount of the students have not attempted the test only. Why? Let's see variety of the reasons now.
I attempted.
You did. Khadija did. Ikhra, you also did. Mariam Lohani, you also did. I can't see your face. It's blank. Are you there in the class?
Ma'am, I also attempted.
Who? Who?
Ikra.
Yeah, I said you did. Yeah, Ikhra also attempted. I have checked your paper as well. So I checked all of your paper. Okay. Now for all of those. Okay, yeah. Yahya, why didn't you attempt the test? Hmm? Many students are not there in the class also. Okay, so while I was checking your paper, students, there is one common thing that I observe in all of your answers is that, okay, I understand that 2 marks question, what more could you write in it, right? We think like that. But remember, always remember, you can say the more you can explain your answers in a better way, the more you are likely to get higher marks in that question. For example, if the question contains of 2 marks, okay? Now, if you write just one long line and that is it, just one line, purely like a proper answer. So I don't think any teacher is going to give you marks. Even for 2 marks, you are supposed to write, you can say 2 lines at least, if I talk about that. And apart from writing the 2 lines, if the question comes along with examples, please mention that, right? And there was this question about this thing, Down syndrome. So only one student has written that it happens because of the trisomy of chromosome number 21. Only one student has written. Yes, you are the one, Khadija. Only one student has written this. Rest, all of you have written it is a chromosomal disorder. This happens in it. But why is it happening? And why we are classifying it in a chromosomal disorder? Because the trisomy is happening. And you have explained it very nicely, Khadija. But the thing again lacking in your answer is that what are the physical things that you can see in a person affected with the Down syndrome? Because that was a 5 marks question, right? So in 5 marks question, you're supposed to give the story. Like you are supposed to believe that the teacher does not know anything. And in your answer, you have to write the answers in such a way that you're teaching the teacher. Hmm? And yes, one more thing. Okay. There was this question about what are the examples of Mendelian disorders. Only 2 diseases are there. Only sickle cell anemia and only hemophilia. Rest were all like other diseases. Where did it go? So pay attention. And like how much time do you get per subject during your tests? How much time do you get?
I only had bio that day.
Oh, you only had bio. And as far as the bio was concerned, how much time did you get?
Like 1 hour, 15 minutes, like that.
Okay. So I think that's sufficient for uh how many questions were there? 15 questions, 13 questions.
13.
13 questions. I think that's sufficient. Two 5 markers were there, and rest all were 2 mark questions, right? Ikra, you wanting to say something? You are constantly
No,
unmuting
no,
yourself.
no.
Okay.
No, no,
All
ma'am.
right. All right. See, students, the reason why I discussed the paper and how to write the question is because it is going to benefit you when you write boards exam. You know, in boards, they don't pay attention to every single thing that you've written, but they're going to see the first half of the answer. Like, have you written facts? Because they also know students make stories. And if you don't make stories at all, so how will you get marks? And by stories, I do not say that write anything in it, right? Appropriate things. Teach the teacher. That is what I'm asking from you. Okay. Now, coming on to this particular thing. Now, we were talking about template dependent fashion. Now, template dependent means this sequence that you're seeing over here. The same sequence is provided of the DNA during the process of transcription. Why? Because the whole entire process is DNA dependent, right? So while it is DNA dependent, anything, whether the new DNA will be synthesized in the case of DNA replication or whether the new RNA will be synthesized, it will happen complementary to the sequence that had already been existing, isn't it? So this is the reason why the process is known as template dependent, because it is dependent on this. New codes or new base pairs are not going to be made by themselves. Okay. So the ones that have already been there, against them, complementary to them, the new sequence will be provided. Clear to you all? That is why it is known as template dependent. Now, students, many a times this diagram will be given in the question and they'll ask you to describe any one of the steps, or they'll ask you like which process is this, whether it is prokaryotic, whether it is eukaryotic. They'll ask you variety of questions related to the diagram also. They'll not ask you to draw it, but see, now since this chapter, like whenever this chapter will get over, so you are to expect that this chapter will be given in the test over here. I'll give it. Now, if in the full marks question, like 5 marks question, if I give transcription, no matter whether I give it for eukaryotes or prokaryotes, you're supposed to draw this. Okay? You're supposed to draw this whole entire thing. It doesn't have to be color coordinated or perfect, but you draw this just so that I will understand that you have understood the topic. Okay? Clear to you all? Okay, now moving forward.
Ma'am.
Okay. Now, students, your videos are on. At least nod. At least have the decency that the teacher is watching. I'm like, did you understand? You're like, I'm like, did you understand? You're like, still the same. At least nod. At least acknowledge that I'm here in the class. Okay. I am just so excited for this chapter to be over now. Okay. Now, the RNA that is formed at the end of the transcription, it is mRNA. Now, the most unique thing is that the transcription and the translation in the case of prokaryotic organism takes place in the cytosol or the cytoplasm, because again, there is no compartmentalization. The whole entire process of transcription is same when we talk about the eukaryotic organism. Just there are some additional steps that happen and there are complexities. Why? Because the organism itself is complex now. So talking about, there are 3 different types of RNA polymerases present. Each of them are DNA dependent only, and their functions, or they transcribe or do the transcription of different kinds of RNAs, right? So these are the ones that we have discussed. And this hnRNA is what my question was. The hnRNA is known as heterogeneous nuclear RNA. And this is the precursor of mRNA, meaning first the hnRNA is formed. Then this hnRNA undergoes several processes of, you can say, modification, and then it becomes the final mRNA. Now, what happens after transcription is completed, students? After transcription is done, what happens after that?
Translation.
Translation. What is the layman term of translation? Can anyone say? Siddhika, can you tell me what is the layman term of translation? Amna? Like translation can also be called as dash. What is the dash? Okay, Mariam Lohani, you tell me. Translation can also be called as? Are you there in the class? Okay, who will say? Who knows the answer?
Is it protein
Ma'am,
synthesis?
Yeah, it's protein synthesis. If you say protein synthesis or you say translation, it's the same and exact thing, right? Now, yeah, so during the process, or you can say additional processes that takes place in the case of eukaryotic organism is splicing, which is the removal of introns. Then capping and tailing also takes place. Now, in capping and tailing, you people have to remember the technicalities as to at what point the methylene guanosine is added, at what point the adenylate residues are added, how many adenylate, this is also asked, like how many adenylate residues are added. So all of these things you have to keep in your mind. So now moving forward, we'll talk about the features of the genetic code. Now, genetic code, you people understand what is genetic code? Everyone understands that,
Yes.
right?
Yes, ma'am.
This G, C, T, A, the whole entire thing that we write, that is the genetic code. Now, remember that you people remember frame shift mutation from genetics chapter? Frame shift mutation. How many of you remember? Come on, you don't hate that chapter, right? So how many of you remember what is frame shift mutation? Okay. All right. Talking about the features of genetic code. Remember that the genetic code is always read in the form of triplet. Triplet means that it is read in three, right? Whenever anything was added, the frame shifted. You people even answered in the class that way. All of you did answer. Anyway, so it is read in the form of triplet. For example, if I have written G, C, T, A, T, A, G, C, and so on, it will be read like this. Three of them will be read together. Hmm? So this is what is known as the triplet reading, or you can say the feature, that is, we always form the genetic code in the form, like in a triplet manner. Three nitrogenous bases in it. Now, the second thing is that they have a comma-less nature, meaning the codon is read without any punctuation. For example, AUG is there, so there will be no stopping in the middle. AUG will be read as such throughout. Then the genetic code is degenerate, meaning some amino acid, they are coded by more than one code. Degenerate means it's not very specific. It doesn't mean that there is one codon which will code for one amino acid. No. There are some codon that are like, there are some amino acids that are coded like by more than one codon. Example is serine, leucine, and arginine. These three. Then the fourth is that it is a universal code. Universal meaning it is applicable to every single organism. For example, if triple U codes for phenylalanine, then it codes for phenylalanine in bacteria as well as up till human beings. So it doesn't matter if the organism is prokaryotic or the organism is eukaryotic. It is universal for all. Now, the initiator codon or the start codon, AUG, this codon is responsible for coding for the amino acid methionine. But apart from that, it also acts like an initiator codon, meaning it will start the processes. It will start the process of any kind of reaction. So it is an initiator codon also, and it codes for methionine also. Then talking about the stop signals or terminator codons. UAA, UAG, and UGA. They have their own name, ochre, amyl, and opal. So they don't code for any kind of amino acid. They are stop codons, meaning wherever they will be present, the whole entire process will stop then and there. And they are also known as nonsense codon because they don't code for anything at all. For example, see, here I've written this particular set of codes. So AUG is the initiator codon, right? It starts the process. Then UUU, it will make its phenylalanine. CUU will make something else. Now in the middle, at the end, there is GUA. And second to it, meaning second last, is UGA, which is opal, one of the stop codons. So the codon, this one will be red, this one will be red, and this one will be red. But because of the presence of a stop codon over here, the process will stop after this, meaning this one will not be red. The whole entire process stops. The reading of the codon also stops because it is a stop codon. Clear to you all?
We stop at UGA.
Either at UGA or UAG and or UAA. If either of them is present during the codon area, or you can say the whole entire template, the process or the reading will stop. Meaning it stops here only. After that, it does not get read. Okay? Clear to you all? Now see students, I'll tell you one of the most important thing. Do you see that I've marked stars everywhere? I hope that tells you the significance of the stars. These 6 points, learn them by heart. Learn them by heart. They'll be asked in MCQs. They'll be asked in subjective questions. They'll always be asked. This was asked in NEET also. So it is very, very, very important. So you pay attention to this. So since this is very important, does everyone in the class understand this?
Yes, ma'am.
Yes,
All
ma'am.
right. Very good. Okay, now moving forward, let's talk about mutation once again. Sort of a division. Now we know that any change in the genetic sequence of the organism is called as mutation. This shows the effect in the genotype as well as the phenotype of the organism. Now in mutation, there can be the loss of any gene or there can be the gain of a gene. Furthermore, mutation can be point mutation or they can be frame shift mutation. Now, when we are talking about the point mutation, so in this, there is the change of a single base pair, right? Now, when we talk about the point mutation with the help of an example, so we know that the disease which is known as sickle cell anemia, that happens because of point mutation only, because the you can say the glutamic acid in the beta globin chain, it gets replaced or substituted by valley. At which position, students? At which position of the chain does it get substituted? Okay. No one knows. Do you remember that sixth position of beta globin chain? Right? At that position, the substitution of the point mutation takes place, because of which sickle cell anemia happens. And what happens actually during sickle cell anemia? Can anyone explain, like in a very quick manner? Why is it known as sickle cell anemia? Is there anyone in the class who can explain?
The
No one.
shape of the blood cell changes.
The shape of the blood cell changes. Are you answering this question or Google is answering?
No,
You
I,
are?
I remember the diagram.
Okay. Okay. All right. So yes, the shape of the RBC changes because of, you can say, hemoglobin gets less, and the oxygen, or you can say the carrying capacity of the RBC also changes. Now moving forward, talking about the frame shift mutation. Now the frame shift mutation, we know that because of either the insertion or the deletion of three or its multiple bases, resulting in the change in the way the frame is read. We know if any base pair gets added, or you can say it gets deleted, the whole reading frame is changed. Now, this much is clear to you all? Any doubts in the two kinds of mutation and anything about mutation?
No, ma'am.
No, ma'am.
No.
Okay. All right, you people note down till here, then we'll talk about the tRNA and its structure. What is the last thing that you people had written in your notes? Hmm?
Some
What is the last
termination.
thing? Prokaryotic transcription.
We have to start from eukaryote transcription.
Eukaryotic transcription?
Yeah.
Okay.
I'm done.
Everyone's done? Students, are you done? Are you done, students?
Ma'am, 1 minute.
Okay, okay.
Done, ma'am.
Everyone's done?
Yes, ma'am.
What about the others? Are you all done too?
Yes, ma'am.
Yes, ma'am.
I'm done.
Everyone's done?
Yes, ma'am.
Ma'am, done.
Done, ma'am. Done, ma'am.
All of you are done, students?
No, ma'am.
No, ma'am.
Okay.
Ma'am, done.
Ma'am, in that universal code example is UUU.
Yes, that codes for phenylalanine.
Ma'am, in the initiator code on AUG is equals to codes for
Methionine. Methionine. Methionine.
done, ma'am. Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
All right. Now talking about the tRNA. People remember that tRNA is one of the varieties of RNAs that we have. Now talking about the tRNA, we know that it is known as the transfer RNA or the adapter molecule. It constitutes about 15% of the total cellular RNA, meaning in the cell, the 15% of it is constituted by the tRNA. Now, the existence of tRNA, it was noted by Francis Crick. Now, he was the first person to tell us about that tRNA exists. Now, it is also known as soluble RNA, that is sRNA. Now, what is the function? The tRNA reads the code, it binds to the specific amino acids. And there are at least 60 different types of tRNA. We don't have to study all those types. Now, talking about the structure of tRNA, so there are two structures that have been proposed for tRNA. One is the inverted L-shaped structure, and the other one is the clover leaf shaped structure. The actual structure of the tRNA is the inverted leaf structure, but the secondary structure is the clover leaf. The one that is given in your book, that is the clover leaf shaped structure. Now, when you look at that structure, you will find that there are three loops present in the tRNA. The first one is the amino acyl synthetase binding loop, or you can also call it as DHU loop, that is dihydroxyuridine loop. So it is the first loop from 5' end. Then the second one is the ribosomal binding loop or T-Chi-C loop. It is the first loop from the 3' end. So you'd see that one is like this, and then there is a smaller one like this. This is the shape that might be given in your book. So one starts from the 5' end. So the first one that we're talking about is the amino acyl synthetase binding loop. This is from the 5' end. The other one comes from the 3' end. So this is the structure that you have to see in your book. Now the third is the anticodon loop. Now the anticodon loop, it has base pairs, like base complementary to the code, and it also has an amino acid acceptor end, meaning this part of the tRNA, it is responsible for accepting any kind of amino acids that come. And remember that tRNA is one of the most important RNA with respect to the translation process. tRNA is majorly involved in this process. Now the inverted L-shaped structure that we talked about, which is the actual structure, that is the 3D structure of tRNA. Whereas the clover leaf model is just a model to study it, so it is a 2D structure. So many a times a question like this can also be asked, like which is the 3D structure of the tRNA or which is the 2D structure. Now see, this particular structure is the inverted L-shaped structure. This is the 3D one that we do not have to know about or do not have to study about. But this structure, see this. This is the one that has been given in your book, and this is the one that you're supposed to study. So from the 5' position, there is one loop. From the 3' position, there is another loop, right? So each of these loops, they have their own different functions to perform. And always remember that the tRNA molecule, they are very helpful in the process of translation. They play a very major role. Now talking about the translation process. Polymerization of amino acid to form a polypeptide. Polymerization means when several amino acids, they come together, meaning amino acids are the monomers. So they are coming together and they are forming the polymer, that is the protein. And we know that translation is the synthesis of proteins. Where does it take place? So in the protein factory, that is the ribosome. Right? You people remember ribosome? It is the protein factory. Now, Ribosomes of the eukaryotic organism, it is 80S and it has two subunits. One is the larger subunit, which is 60S, and one is the smaller subunit, that is 40S. So mostly if we are talking about translation, so we're going to talk about the eukaryotic translation because it is properly defined. Now the ribosomes, they have two sites for binding of amino acyl tRNA. There is P site present in the ribosome and then there is A site present in the ribosome. So in the ribosome only there are two sites present. And each of these two sites, they're extremely important again for the protein synthesis. Now when the small subunit encounters or comes in contact with the mRNA, the process of translation starts then. Meaning this is the larger subunit, this is the smaller subunit. As soon as the smaller subunit will come in contact with the mRNA, the process of translation will start. Now the amino acids, they are joined together by peptide bonds. So always remember that in the case of proteins, peptide bonds or polypeptide bonds are present. Protein is a polypeptide and it has peptide bonds present in it. So yeah, so first you people note down till here. Then we'll go forward and talk about what are the various steps that take place in the translation process. We're almost near to the end of the chapter. Okay, can you see my screen, students?
No, ma'am.
Now?
Yeah, no.
Yes.
Okay, yeah, now you can note down. Done, students?
Yes, ma'am. Done, ma'am. Done, ma'am.
If you're done,